So what I want to do is tell a quick story, a microbiology story, but don't worry, it's a really interesting organism. So the name of this microbe, of this bacterium, for those of you who are physicists and so forth, this small bacterium is Listeria monocytogenes. And Listeria monocytogenes, it's an organism that lives in soil and water and so forth. Sometimes it gets into food, and sometimes if people eat that food, they can get very sick. And specifically, people who are aging, older people who are pregnant, and I'll explain why it's particularly dangerous for people who are pregnant. But the important thing about this bacterium is that it's a uniquely human infection. It doesn't infect mice, it doesn't infect horses, it doesn't infect cows and rabbits and so forth. It only infects humans. So staying in this little science lecture, mini science lecture, one of the reasons it's so interesting is that it has this ability to enter this big green. Do you have a pointer on this? Yeah.
Yeah, there's a red dot.
There. So this rather large green object is a cell, a macrophage, or some kind of cell. And here's our bacterium infecting the cell, breaking into the cell. It's in the cytoplasm now. It's able to jump out of a membrane that usually contains it, and then it harnesses the actin inside the host. So now here's a bacterium collecting actin filaments. We all know what actin filaments are. Inside the cell and using them to move across the cell and go into the neighboring cell. So let me just explain that if you take a petri dish of mammalian cells, eukaryotic cells, just epithelial cells, for example, and drop one of these bacteria in the middle of the plate, it will completely destroy the entire plate in a few days. And it does this by moving from cell to cell and bursting the one behind it, going into a new cell, bursting the one behind it. So it will take over the population. It does this in the human brain. It crosses the blood-brain barrier, and it crosses the placental barrier, and so it can cause very serious disease in which a fetus is destroyed. It's a terrible organism. But remember, it can't be studied in mice because it doesn't infect mice. Right? So how do you work on this? You have to work on this in the lab. This is some scanning hands. And some photographs of how the bacterium, this is the bacterium, this little yellow-orange thing is the bacterium, and here it is harnessing and picking up all the actin and moving around. So it's also a beautiful bug. So here's this problem. We can't study, we don't have a mouse model to study the disease of listeriosis. But in 2007, a group in Germany figured out a way to change the genetic code so that now the bacterium can infect mouse cells. Right, so they did this experiment, and it was published in the journal Cell, very famous. And they thought very carefully how to do it. They were able to predict exactly what genes and exactly what sequences in the genes to change so that now this bug can infect mice. And these are the changes. We don't need to see them, but there they are. And so Cell, the journal Cell was very happy about this, and they bragged and put a little paragraph in the front of the journal to say this is cutting edge, leading edge research. These scientists have now figured out how to manipulate the human pathogen Listeria monocytogenes to extend the host range to mouse. And this way, they've created a versatile animal model of human listeriosis. So Cell was very happy. Researchers in this field were very happy. Now they have a mouse model. However, the Washington Post was not happy. So the Washington Post interpreted this technological advance, this scientific advance in microbiology and infectious disease as a double-edged advance that could deepen scientists' understanding of human disease but also speed the development of novel bioterrorism agents. Right? So this is a really very clear description of how we think about basic research designed to help humankind to cure diseases that has a potential for misuse. So just a very short exercise. Should I make it? Yes. Very short exercise. No reporting out. This is just the groups to talk among yourselves. can you name, are there some experiments that you would not do because the results could be misused to create a weapon, for example? And I know we have some people not in biological science, so that would be great to think about physics and other things as well. Questions about that? No, it's going to be just a talk, and not even reporting out. Just want everybody to start thinking this way.